Today I'm presenting a primary research article on T lymphocytes and cytotoxic astrocyte blebs correlate across autism brains. So just a little background on autism spectrum disorder, also known as ASD. It manifests in early childhood and is diagnosed based on behavioral deficits. Core symptoms include social communication challenges as well as restricted repetitive behaviors. So the researchers were looking at ASD human postmortem brain tissues to try to gain an understanding of some of the pathological processes that underlie this disorder. So they utilized a number of different techniques. The first was histology and immunohistochemical staining, in which they stained with a hematoxylin and eosin stain and a Luxol fast blue. Next, they performed an automated lymphocyte detection and segmentation procedure in which they photographed 3 blood vessels with the most abundant perivascular lymphocytes in each of the following brain compartments: gray matter, white matter, or leptomeninges. Next, they did an automated white matter perivascular space segmentation procedure where they looked at 10 white matter vessels surrounded by the largest space intervening between the vessel wall, and white matter parenchyma were selected. Next, they did automated trichrome perivascular collagen segmentation, where they looked at 6 blood vessels in white matter surrounded by the largest amount of blue-stained perivascular collagen among all blocks for each ASD in control cases. Lastly, they did immunohistochemistry, imaging acquisition, and analysis, where they looked at 10 of the white matter blood vessels with the most abundant perivascular GFAP debris, which is known as glial fibrillary acidic protein, across all of the blocks containing cerebral cortex in each case of ASD and control cases to quantify this debris. So now we're going to go into the results of this experiment. So the first thing they found was that there was damage of CFS brain barrier by astrocyte processes in ASD. So figure 1 shows that perivascular membranous blebs stained with multiple astrocyte markers, and they also co-localized with CD8 plus T cells in ASD brains. Specifically, the S100B and the ALDH1L1 were 2 astrocyte markers that stained in the perivascular membranous blebs. Next, they looked at CD8 cytotoxic T lymphocytes are increased in autism spectrum disorder. Specifically, figure 2, shown here, shows that lymphocytes are increased in the cuffs around a subset of vessels, and their numbers correlate to the quantity of perivascular GFAP plus membranous blebs across ASD cases. Figure 3 elaborates on this and shows that perivascular cuff lymphocytes are increased across brain regions as well as compartments and ages in ASD. Lastly, figure 4 shows that CD8 plus cytotoxic T lymphocytes were predominant in the focal perivascular cuffs in ASD brains. The next finding they focus on is how perivascular space is increased and has a more jagged contour in autism spectrum disorder. So specifically, they found that perivascular space, like lymphocyte numbers, was increased around vessels in ASD relative to control when comparing these top 10 vessels. Next, the dots represent a jaggedness index for the area surrounding a single vessel, and the aggregate differences between autism and control counts was statistically significant. The last thing they looked at was how perivascular fibrosis was increased in autism spectrum disorder. Specifically, adventitial perivascular collagen was increased at these top vessels from ASD relative to control. The amount of perivascular collagen in white matter for each measured vessel in every case normalized to the area of the vessel lumen and was higher across autism cases compared to controls. Again, the aggregate differences between autism and control counts are statistically significant. So some concluding thoughts by the authors. They believe that the increased collagen deposition and expanded perivascular spaces with jagged contours in ASD suggest a destructive effect of the elevated cytotoxic T lymphocytes. In addition, the number of perivascular lymphocytes and the amount of GFAP debris correlated across ASD cases, a finding that suggests that cytotoxic T lymphocytes may generate the astrocyte membranous blebs. They also suggest that they may be generated by a targeted attack of astrocyte processes by cytotoxic T lymphocytes, specifically at the glial limitins surrounding blood vessels and other CFS brain barriers. Lastly, CD8 plus T lymphocytes, infiltrates, and cytotoxic astrocyte membranous blebs represent a new cellular process not previously reported in studies of ASD neuropathology. For future research, the researchers are interested in testing if the maternal, fetal, or autoimmune antibodies found in the serum in some cases of ASD co-occur with the CD8 plus T lymphocyte immune cell infiltrates and astrocyte membranous blebs, or if they represent a separate autoimmune subtype of ASD.